In the next two classes, we're going to talk about anaerobic digestion. When the biodegradable COD in a waste stream is very high, the transfer of oxygen becomes a limiting factor. Anaerobic processes become ideal treatment selections as they do not need supply of electron acceptor and convert organic carbon to methane gas, which can be recovered as an energy source. Therefore, anaerobic digestion has been widely used by utilities to treat their high-strength solid waste streams, such as primary sludge and waste-activated sludge. They're also adopted by various industries to treat their high-strength industrial wastewater, and also gain momentum in treating food waste collecting from food manufacturing and processing industries, grocery stores, restaurants, and households. The biochemical reactions in anaerobic digestion process are multi-step in nature, meaning that as the reaction cascades down, the product from an upstream reaction performed by one group of microorganisms becomes the reactant for a downstream reaction performed by a different group of microorganisms. This makes AD process very complicated. Moreover, some of the products can be inhibitory to its own degradation and others. We can roughly categorize the complex organic matter into carbohydrates, proteins, lipids, and inerts. Inerts do not participate in any of the following reactions. For hydrolysis by extracellular enzymes, carbohydrates are broken down into simple sugars, proteins into amino acids, and lipids into mostly long-chain fatty acids. Through glycolysis, Stickland reaction, and beta oxidation, simple sugars, amino acids, and long-chain fatty acids are degraded to volatile fatty acids like the 5-carbon valerate, 4-carbon butyrate, 3-carbon propionate, and eventually to the 2-carbon acetate. These processes are called acetogenesis and acetogenesis, or generalized as fermentation. Hydrogen gas is generated through these reactions. Finally, the acetate is converted to methane through acetoclastic methanogenesis, while the hydrogen gas and carbon dioxide are also converted to methane gas through hydrogenotrophic methanogenesis. These different processes are performed by many different complex microbial communities that has different optimal growth conditions, yet are interdependent on each other. To make things more complicated, many of the products of these processes can be inhibitory to other processes as well as its own consumption. For example, ammonia is produced from a degradation of proteins and amino acids through a modification. Free ammonia can diffuse through cell membrane and protonate, disrupting proton balance and inhibit various bioreactions. Free volatile fatty acids, especially acetic acid, not only disrupt the proton balance at cellular level, but also lowers the pH in the reactor, reducing the growth rate of pH-sensitive species like acetoclastic methanogens. Long-chain fatty acids are hydrophobic and are thought to bind to the cell membrane lipid bilayer, blocking substrate mass transfer. The beta oxidation of the 4-carbon butyrate and 3-carbon propionate to form acetate is thermodynamically unfavorable, with a positive delta G naught. A low enough hydrogen concentration is needed to make this reaction move forward. But in the meantime, hydrogen is the substrate for hydrogenotrophic methanogens. Hydrogen concentration directly affects their growth rate. A delicate balance needs to be maintained to ensure steady operation. Any system perturbation needs to be addressed in a timely manner, or it can result in the accumulation of inhibitory intermediates, which can snowball into system failure. Because the anaerobic microorganisms are slow-growing due to thermodynamic limitations, restarting the reactor can be time-consuming and costly. Because the particulates like carbohydrates, proteins, and lipids are too large to be uptaken directly by microorganisms and utilized as electron donors. They are first hydrolyzed to smaller molecules by extracellular enzymes excreted by hydrolytical bacteria, fermenters, and perhaps methanogens. Hydrolysis is not a redox reaction, so there is no loss of COD during this process. Because of the large size, the specific surface area of the particulates that the extracellular enzymes can bind to is low, making hydrolysis a rate-limiting step for the anaerobic digestion of high solid waste streams. Acidogenesis and acetogenesis, which are also referred to as fermentation process, are conducted by fermentative bacteria like Clostridium species. These are three reactions representing the fermentation process. Glucose can produce 2 acetate, 2 CO2, and 4 hydrogen gas, or 1 butyrate, 2 CO2, and 2 hydrogen gas. Or it can consume 2 hydrogen gas to produce 2 propionate. The butyrate produced from equation 2 can be further degraded through beta oxidation to produce 2 acetate and 2 hydrogen gas, making the net production the same as equation 1. The same applies to the propionate produced in equation 3. But because the delta G naught for beta oxidation of butyrate and propionate are positive 48.2 kJ per mole and 76.2 kJ per mole respectively, these reactions are not thermodynamically favorable under standard conditions. Hydrogen concentration needs to be sufficiently low to make these reactions move forward spontaneously. During hydrogenotrophic methanogenesis, hydrogen is consumed to produce methane. If the reactor is well balanced and we have a healthy hydrogenotrophic methanogen population that helps keep the hydrogen partial pressure low, equation 1 should dominate. But if too much substrate is fed and hydrogen consumption rate by hydrogenotrophic methanogens cannot keep up with the hydrogen production rate, there will be more accumulation of propionate and butyrate in the reactor. So, to make the reaction happen spontaneously, delta G naught needs to be less than 0. Can you calculate the highest partial pressure of hydrogen that can make this happen based on this equation? We can reasonably assume that the concentration of propionate equals to the acetate and the partial pressure of CO2 is 0.3 atm. And can you do the same for butyrate with the same assumption? Those microorganisms that convert butyrate and propionate to acetate are called centrophic bacteria, meaning that they utilize products and metabolites produced by others as substrates. They are generally very slow growing due to the thermodynamic limitations, although they play very important roles in anaerobic digestion by providing substrate for methanogens and avoiding the accumulation of inhibitory intermediates like propionate and butyrate. Because the centrophic bacteria that consumes butyrate is faster growing and have wider substrate spectrum than the propionate consuming ones, we generally see accumulation of propionate first in the digester that is overfed, followed by the accumulation of butyrate and hydrogen. During the degradation of amino acids, ammonia is produced through a modification. Ammonia is important not only because it is an inhibitor that can disrupt proton balance and deplete intracellular potassium, it is also an important buffer that keeps the pH stable, as the production of volatile fatty acids can decrease the pH, which can lower the growth rate of pH-sensitive methanogens. The final step, methanogenesis, has two major pathways: acetoclastic methanogenesis that converts acetate to methane, and hydrogenotrophic methanogenesis that converts hydrogen and CO2 to methane. Although over 2/3 of the methane is produced from acetate in anaerobic digestion, very few methanogen species can do this. we know these two major genera, Methanosarsina and Methanocyaeta. Even though Methanocyaeta has a lower mu max compared to Methanocyaena, its K sub S value is low, making it the dominant acetoclastic methanogen in typical anaerobic digesters, which have low acetate concentration. As for hydroneutrophic methanogens, they are responsible for 1/3 of the methane production by utilizing the hydrogen gas produced by the syntrophic bacteria from degrading fatty acids. They also help reduce the hydrogen partial pressure, which helps syntrophic bacteria utilize their substrates. It is a mutually beneficial relationship. Note that methanogens are archaea, the third major branch on the tree of life besides bacteria and eukarya. One major benefit of anaerobic processes, not just anaerobic digestion, are the low sludge production due to the low yield. This can save solid disposal cost, a major cost next to aeration cost by utilities. It also saves aeration cost as oxygen is not needed in anaerobic reactions. Actually, energy can be recovered from waste stream electron donors as methane. Anaerobic processes can also remove toxic xenobiotics that are difficult to remove aerobically. For example, chlorinated organic compounds can be removed through reductive dehalogenation. Anaerobic processes also have low nutrient requirements. But as a result of the slow growth rate of anaerobic microorganisms, a long SRT is often required for anaerobic processes. If there is no separation of SRT and HRT, it will require a large reactor footprint and high cost. It is often the case because it is difficult to settle anaerobic biomass, unlike aerobic ones. New technologies like membrane bioreactor and various attached growth reactors have been developed to tackle this. But they also have their own issues, such as membrane fouling for membrane bioreactors. The slow growth rate also means long startup period and slow recovery after reactor upset. Anaerobic microorganisms, especially methanogens, are sensitive to environmental changes like temperature and pH. Typically, the pH needs to be maintained between 7 and 8, and the temperature should be either mesophilic, which is around 35 degrees Celsius, or thermophilic, which is around 55 degrees Celsius. Because of the pH requirement, diluted or carbohydrate-rich waste streams do not have sufficient alkalinity for anaerobic treatments. If sulfate is present in the waste stream, it can be reduced to sulfide during anaerobic processes, causing corrosion and odor issues. Finally, neither nitrogen nor phosphorus can be removed via anaerobic processes alone. Although NMOX reaction happens anaerobically, it also requires partial oxidation of ammonia to nitrite.